Hey everyone, and welcome to BioBusters. In this video, we'll cover everything you need to know for Subtopic 1.1, Introduction to Cells, of Topic 1, which is Cell Biology, of the IB Biology HL curriculum. I'll begin by introducing the concept of cells and their principles. We'll also talk about atypical cells, the functions of life, uni and multicellular organisms, cell differentiation, and emergent properties. We'll then move on to stem cells, stem cell therapy, and the ethical debate surrounding their use. Lastly, we'll cover the concept of surface area to volume ratio of a cell, and we'll do some magnification calculations. Let's get started. First off, what is a cell? Well, it's the smallest unit of life, but this is quite a simple definition. So, in 1838, Theodor Schwann, Matthias Schleiden, and Rudolf Virchow created the cell theory to explain the three most fundamental principles of cells. The first one is living organisms consist of one or more cells. Secondly, the cell is the smallest unit of life. And third, all cells arise from pre-existing cells. However, there are some cells that do not follow all of these principles, and they're known as atypical cells. Let's look at three examples. The first is the striated muscle cell. This cell is very long and it has multiple nuclei surrounded by one plasma membrane, so each cell unit is multinucleated. Hence, this challenges the idea that cells always function as autonomous units with one nucleus. The second is the giant algae. This is a unicellular algae, and it challenges the idea that larger organisms are always made of many microscopic cells, and cells must be simple in structure and small in size. And third, there's the aseptate fungal hyphae. These cells form long threads, hyphae, with many nuclei and have no dividing cell walls, so they challenge the idea that a cell is a single unit because they have many nuclei, are very large, and have one long cytoplasm. Now, how do we know these cells are or form part of living things? Well, we have to look at the functions of life. These are metabolism, growth, response or sensitivity, homeostasis, nutrition, reproduction, and excretion. Metabolism is a set of life-supporting chemical reactions that take place within cells of living organisms. Growth is the increase in size or shape that occurs over a period of time. Response or sensitivity is the reaction by the living organism to changes in the external environment. Homeostasis is the maintenance of an internal environment by regulating internal cell conditions. Nutrition is the intake of nutrients and the exchange of materials and gases with the environment. Reproduction is the production of offspring, either sexually or asexually, to pass on genetic information to the next generation, and excretion is the removal of waste products of metabolism and other unimportant materials from an organism. For something to be a living thing, it must be capable of carrying out all of these life functions. There are two classifications for all living things: unicellular, which are single-celled organisms, and multicellular, organisms with many cells. Examples of unicellular organisms include the paramecium and the Cleidomonas. The paramecium is a protozoa that lives in aquatic environments, such as stagnant ponds, while the Cleidomonas is a unicellular green algae that can live in soil, fresh water, oceans, snow, and mountain drops. For the IB, you must be able to say how each of these organisms performs the life functions. So let's go over that. First, they both perform metabolism in the cytoplasm, and it is catalyzed by enzymes. To perform growth, the paramecium, a heterotroph, ingests nutrients and increases in size. However, the Chlamydomonas is an autotroph, so it performs photosynthesis and absorption of minerals to increase in size. To perform response, the paramecium uses the cilia to propel it in response to the environment, for example towards warmer water. However, the Cleidomonas has an eye spot, which senses light changes, and then the flagella moves it towards brighter regions to increase its photosynthetic rate. To perform homeostasis, they both use osmoregulation to keep the water balanced by collecting excess water in contractile vacuoles and expelling it through the plasma membrane. To perform nutrition, the paramecium, since it's a heterotroph, uses the vacuole to engulf food particles, and it also eats microorganisms such as bacteria, algae, and yeasts. However, the Chlamydomonas, being an autotroph, uses the chloroplast and photosynthesis to produce its own food. To reproduce, the paramecium can perform sexual or asexual reproduction through binary fission. However, asexual reproduction is more common. However, in the Chlamydomonas, sexual and asexual reproduction are both used. To excrete, the paramecium removes solid waste via its anal pore, and liquid wastes are pumped out via contractile vacuoles. However, the Chlamydomonas uses the whole surface of its plasma membrane to excrete its waste products. Now, besides unicellular organisms, let's take a look at multicellular organisms. These can include animals, plants, and fungi, and rather than only having one type of cell, they have many. In humans, for example, some different types of cells are smooth muscle cells, fat cells, red blood cells, and nerve cells. But how do these cells become different? Here's where we look at stem cells and cell differentiation. A stem cell is an undifferentiated cell of a multicellular organism from which certain other kinds of cells arise by differentiation. They have two properties: self-renewal, meaning they can continuously divide and replicate indefinitely, and potency, meaning they can differentiate into specialized cell types when given the right stimulus. It's important to note, however, that not all stem cells can give rise to all body cells. There are four types of stem cells: totipotent, which can form any cell type, including placental cells, and give rise to complete organisms, for example, a zygote; pluripotent, which can form any cell type, for example, embryonic stem cells; multipotent, which can differentiate into few closely related types of body cells, for example, umbilical stem cells; and unipotent, which cannot differentiate but can self-renew, for example, muscle stem cells. To differentiate, stem cells undergo specialization, a process by which unspecialized stem cells develop into cells with a more distinct structure and function to carry out a specific function in the body. This occurs due to the expression of different genes. So, to evolve from uni to multicellular organisms, cells clumped together, and over long periods of time, they became simple multicellular organisms. The organisms grew larger as they were no longer limited by the size of one cell. Differentiation occurred and emergent properties surged. Emergent properties only occur in multicellular organisms, and they are caused by the interaction of individual components to produce new properties that its individual constituent parts do not have. With stem cells come a lot of advantages in the biomedical field. This can occur with stem cell therapy, wherein stem cells are used to replace damaged or diseased cells with healthy, functioning ones. This procedure can be used to treat many diseases, among them Stargardt's disease. This is an inherited form of juvenile macular degeneration that consists of progressive vision loss until blindness. It's caused by a gene mutation in the ABCA4 gene that causes retinal photoreceptor cells to degenerate. So, using stem cell therapy, dead cells in the retina can be replaced with functioning ones. Sadly, however, there are some ethical issues of using stem cells depending on their source. If they are derived from embryos, they have the greatest stem cell yield, but it requires destruction of a potential living organism. If they are derived from the umbilical cord blood, it may be effective, but they need to be stored and preserved at cost, so it raises issues of availability and access. And if they're derived from multipotent adult tissue, they are effective sometimes, but the application scope is limited. Overall, using stem cells from embryos are found to have the greatest advantages. This is because it can help eliminate serious diseases, it involves easy transplants and don't require the death of another human or inflict any pressure on normal body functioning, and if they're harvested at an early stage, the embryo has no developed nervous system, so it's likely that it feels no pain. Two artificial stem cell techniques include somatic cell nuclear transfer, also known as SCNT, and nuclear reprogramming. SCNT is the creation of embryonic clones by fusing a diploid nucleus with an enucleated egg cell. The advantages are that they are indistinguishable from embryo-derived cells. However, the disadvantages are that doing this involves the ex vivo creation of embryos, so more than needed are made. Nuclear reprogramming involves inducing a change in the gene expression profile of a cell in order to transform it into a different cell type. The advantage is that the stem cell is autologous to the adult donor. However, the disadvantage is that it involves the use of oncogenic retroviruses and transgenes, so there's a higher risk of health consequences. Now, there's one more important concept about cells that we need to cover, and that is the surface area to volume ratio. A larger surface area of a cell means more material movement in and out of the cell. A larger volume of a cell means a lower rate of metabolism. When a cell grows, its volume increases more than its surface area because volume is cubed while surface area is squared. Sometimes, the surface area of a cell is too small compared to its volume. When this happens, not enough of the necessary molecules can get in, and not enough waste, including heat, can get out of the cell. So the cell divides into two smaller cells. Examples of cell adaptations to increase the surface area are the folds in the small intestine. With the folds, more surface area is exposed to the digested food, so more food absorption can take place. There's one last thing the IB wants us to know for this topic: calculating the magnification of a cell. To do this, we use the formula magnification equals image size with the ruler divided by the actual size according to the scale bar. Let's do an example. We are asked to calculate the magnification of the following electron micrograph image. It's given that the actual size of the image is 0.5 micrometers. Let's say we used a ruler and found that the line measures 5 cm on paper. We can then use the formula to find the magnification by dividing the image size by the actual size. But first, we should convert everything to the same units. Let's use meters. So 0.5 micrometers is equal to 5 times 10 to the power of negative 7 meters, and 5 centimeters is equal to 0.05 meters. So using the formula, we get that the magnification of the image is 100,000x. And that's it for Subtopic 1.1 Introduction to Cells of the IB Biology curriculum. Thanks for watching!